like.
Good afternoon, one and all. Present here for the fourth session of the webinar series on fermentation technology, organized by the Department of Biotechnology, Guru Nanak College, Velachery, Chennai. I deem it a privilege to request our Dean Research, Dr. J. Jayanti Madam, to welcome the participants. Madam, please.
A very good evening to all the participants who have gathered here. On behalf of our college, I thank the Department of Biotechnology for taking such a wonderful initiative in bringing out such a wonderful course on fermentation technology, which is the need of the day. As a Dean Research, it is my duty to tell all the students, whether they are UG, PG or research scholars, that a course on fermentation technology is going to take you a long way in research. So please, all of you, please utilize these sessions. This is a long duration of course. You have 4 days course with 2 sessions per day, and you have some of the wonderful speakers joining us. I think it started out with the IIT speaker, and you can see it is expanding in a very good fashion, and I think it will be very, very useful to all the participants present here. And once again, I'm proud that the Department of Biotechnology, which is one of the well-established department in our college, that has taken up such a wonderful initiative to reach out to the audience, to the undergraduate, postgraduate, and the scholars, to make them understand the importance of fermentation technology and also to inculcate that into their curriculum or in the research work. Thank you. Congratulations to the head of the department, Dr. Bharti Ravi Krishnan, for wonderfully organizing this particular certificate course, and also to the entire team of biotechnology, Dr. Mahendra Kumar, who is technically supporting the entire team, and all the Dr. Amal Raj, Dr. Mahalashmi, who are doing a wonderful work. Thank you, one and all. Thank you for the wonderful opportunity given to me today to join along with you. Thank you.
Thank you very much, ma'am, for your warm words of welcome and appreciation. We hope we will live up to your expectations. Now, I feel very much proud in introducing our speaker of the day. It gives me immense pleasure in introducing our speaker of the fourth session, Dr. Bharti Ravi Krishnan, who is also the convener of this webinar series. Dr. Bharti Ravi Krishnan is currently serving as the head and assistant professor in the Department of Biotechnology at Guru Nanak College, Velachery, Chennai. Earlier, she had served as assistant professor at the Department of Biotechnology in Hindustan College of Arts and Science, Padur, and Kumararani Meena Muttaya College of Arts and Science, Adayar, and as a lecturer at Jaya College of Arts and Science, Thiruvananthapuram. Altogether, she has a teaching experience of 16 years. Madam finished her PhD on crustacean immunology under the able guidance of Dr. M.G. Raghunathan, who is the current principal of Guru Nanak College. As a research person, Madam has served for Biology in School Teachers Forum, Teach the Teachers, organized by TA School, Ambattur. She had presented a talk entitled Sustainable Development Strategies Needed for India Future Pursuit of Happiness in the 104th Rashtriya Vigyan Sancharat Sammelan, which has held during 104th session of the Indian Science Congress from January 2017 at SV University, Tirupati. She had presented a talk entitled World of Microbial Soup at T.S. Narayanasamy College of Arts and Science, Navalur in 2016. She had presented a talk on Preserve Your Feminism to overcome PCOD among students of the Department of Commerce, Hindustan College of Arts and Science in 2016. She had also participated in the demonstrating science experiment to underprivileged students supported by Mambalam Lions Club from 2012 till date. She had also delivered a subject expert talk, Know Your Food, Cosmetics and Your Therapy, organized by the research committee of Kumararani Meena Muttaya College. She had presented a talk at state level conference at Guru Nanak College and delivered a talk on title Science for Indigenous Indian Development. She had also delivered special lecture at Guru Nanak College titled Food Coding for the students of Advanced Zoology and Biotechnology in 2013. She had also presented a concept of tissue engineering in Pop Science Expo organized at Guru Nanak College. She has 26 international and national publications to her credit, and she had won 3 awards. We have such an eminent person with us to deliver the talk on fermentation. Madam, over to you, ma'am.
Thank you, Mahinda Kumar sir. Thank you, Dean ma'am, Dr. J. Jayanti, for appreciating all of us. Thank you so much, ma'am. And now I would like to share my views on overview of types of fermenters and its parts. So when we talk about a fermentation process or a fermenter, let's just look into all these insights. So I've just shown you pictures about pickle in a jar, or mayonnaise preparations, or baking of a cake, or curd, or paneer production, which is very common in India, or this is a de-starched rice, a kind of a food that we always eat in South India, especially in Tamil Nadu. And then we do have a newly fermented coconut-based drink coming up, and which is having a good recognition in the market. All these foods are the examples of fermentation products, which is we do it in our home without any kind of a specific equipment at all. And now these are all the earliest instruments which are being used in our house, or equipments used in our house for to prepare the pickles. So yes, our mothers, our grandmothers, and we also, when we do the picking process, we become the fermentar technicians also. And there are archaeological evidences of fermented products which ages back from 30,000 to 1 lakh years back. And newly, recently, the sculpture of a woman holding a wine vessel is also being identified. Then old vessels in Soviet Union controlled Georgia has been excavated, and from Neophyte era, many apparatus which is used for the beer production, grinding of the barley grains, have been excavated. So with this, let's move on to the timeline of fermentar. So the fermentar design has really taken up its shape from the year 1914 by scientist Chain Weisman, who has developed the fermentar for the large-scale production of acetone. In the year 1930, development of large-scale aerobic fermentar was developed. In the year 1934, scientists Strauch and Stimmett introduced aeration, water, and steam inject flow systems in a fermentar. In the year 1944, scientists D. Bees and Leibman developed the fermentar for the large-scale yeast production. And in the designing of the fermentar, India has also contributed his and in the year 1950, Hindustan Antibiotics Limited, now in Pune, has given the first design for the pilot scale fermentar to the world. And now coming to fermentar. So now what is a fermentar? If you see, it is the heart of this entire fermentation process. So long, the speakers in the first day have given you a clear introduction about different type of fermentation process and their overview and their importance. And in the morning, Madam Dr. Veena Gayatri has clearly explained to you how we have to identify the substrates with which we can go in for designing a medium for the fermentation process. And the medium, the organisms have to be put into a specific vessel, and that vessel or an instrument we call it as the fermentar, which is the heart of any type of fermentation or the bioprocess technologies. A fermentar is usually referred as a containment system for the cultivation of the prokaryotic cells or the eukaryotic cells in a well-specific environment which supports for the product production. Now, let me just give you a small classification or difference between fermentation process and the bioprocess. So when we take up fermentation process, here inside the fermentar, the nutrients or the medium required and the respective cells are being added. So the cells will convert the nutrient into the product, and the product production happens outside the cell. So this is what we mean by fermentation process. When we take the bioprocess, the inside the fermentar, the nutrients, the mediums are added, the respective cells are added, the cell taking the nutrient, and in a cytoplasmic environment, the nutrients or the substrate is converted into the product. So this is bioprocess technology. So the cell here, I mean, it can be a bacterial cell, it can be an animal cell, it can be an algal cell, or it can be a plant cell. Now, coming to the fermentation or the bioprocess technology, we actually use these technologies to produce eco-friendly products. It can be a nutraceutical compound, health supplements, or it can be involved in an environmental degradation process. And this entire process or fermentation process can happen inside a specific closed system, or it can also happen in an open system where the fermentation process can happen inside a contained environment. So I just want to introduce a few points or mention few points about the microorganisms. So first thing is microorganisms are selected from the environment, and then it has been standardized. It has identified whether the microorganism is able to produce the specific product of our interest, and what are all the suitable supportive medium by which the microorganism can grow and also produce the products. So that standardization happens at the laboratory scale, and this is what we call it as a pilot scale. So once the pilot scale has been standardized, we go in for a large scale production of the product, and at the same time, a small batch of cells are harvested and stored as a cell bank. So cell bank maintenance of a specific microbial strain, which is involved in a specific product production, is one of the another important part of this entire fermentation process. So here I've shown you the various pictures of fermenters. So fermenters can come as small models at 3 liters or 5 liters, and it can even be 50 liters to 100 liters at a large scale production at an industry. At Guru Nanak College, Department of Biotechnology, we do have a 3-liter stainless steel stepped type of fermenter with us here. And the main objective of this fermenter is it must support for the product production as biomass or metabolites or enzymes, or it can be even recombinant products, or it can also be a 3D construct. The ideal properties of a fermenter must be that the first thing is the body of the fermenter must be made up of such a material that it is very sturdy in nature, and also it is cheap in cost. It can be a glass or it can be a stainless steel. And the fermenter must support for the large scale production of our products. The fermenter must withstand high pressure or cold conditions or hot conditions during the fermentation process. The fermenter must be supported with ports or sensors to monitor the concentration of the substrate or the inoculant or the temperature or the pH. Now coming to the different type of fermenters, there are different types of fermenters are available with us according to the type of microorganisms we are going to use, or type of medium that we are going to use, or the type of products that we are interested in the production. And we can, according to the research or according to the production that we are interested in, we ourselves can combine 2 or 3 processes of a fermentation process, and we can always construct a fermenter of our interest. But basically, these are all the different type of fermenters that are available and largely used both at a research field and also at the industrial production process. So the fermenters can be classified as constant stir tank fermentar, airlit fermentar, tower fermentar, fixed bed fermentar, packed bed reactors, bubble column tower reactors, vertical tower fermentar, multiple stage fermentar, fluidized bed reactors. And each and every fermentar, what I've mentioned over here, has got its own unique functions. It is involved in the unique product production. See, when we talk about the packed bed reactors, they can be used for the degradation of the environmental toxic waste and also involved in the production of nutraceutical or therapeutic compounds also. Now, coming to the first and simplest type of fermentar is the strict type fermentar. This is widely used fermentar both at an academic, industrial, and also at a research grade. It consists of a cylindrical vessel with a stirrer of the fermentar, and this is connected with the impeller blades. So the movement of the agitator provides the homogeneous medium for the organisms to interact with the substrate and also to maintain the uniform gaseous concentration inside the fermentar. And the stick type fermentar comes with a see-through glass which is supported by a lightning process. So we can literally visualize advantages of this stir type is it is very easy to operate, it is very student friendly, there is a good temperature control, its construction is quite simple, there is low operating cost, the fermentar is easy to clean. Nowadays, a stiff type fermentar comes with an in situ sterilization inbuilt process also. And this stiff type fermentar do not cause shear damage to the cells. The main disadvantage is that is we cannot use immobilized cells or enzymes inside a stirred type of fermentar. It has got a low volumetric productivity, and the major disadvantage of the stir type is due to the continuous stirring process, froth can be generated, and this can sometimes lead to the contamination process or loss of microorganisms present inside the fermentation process also. Now coming to the next type of fermentar, that is the airlet fermentar. So this fermentar actually suits or supports all the aerobic microorganisms has an outermost shell and inner there is a draft column. The air is pumped through the orifice into the draft column. The air that comes inside the draft column is called as a riser, and the air when it comes out of the draft column into the medium, it is called as downcomer. So the riser and the downcomer ratio actually decides what must be the size of this particular airlet fermenter. And the riser and the downer helps in maintaining the homogenized environment for the organisms to utilize the substrate and also to produce the products. Now, there are different type of airlet fermenters as it's shown over here as a draft tube fermentar, where a draft tube is present in the center and there is a cylindrical device septum that is present in the center, which helps in the circulation of the air, or if there is an external loop device based airlet fermentar is also there. So all these in these fermenters, it maintains a uniform homogeneity of the medium and the organisms. And there is no agitator present inside this fermentar. Only the air passage performs this mixing up of this medium or the gas and the organisms. And one type of airlet fermentar that is specifically used to culture the plant cells to produce the metabolites is the bubble column fermentar. Now coming to the packed bed reactors, this is one of a very unique and interesting type of fermentar because this particular design of the fermentar can suit both for the environmental pollutant degradation and also for the production of high value products like therapeutical agents like monoclonal antibodies. So about these a modified form of packed bed reactors, I'll be discussing in the later slides. So now let's look into the packed bed reactors. So in the packed bed reactors, we do have an outermost shell and inner tubes are arranged. So in these tubes, the immobilized cells or enzymes are being immobilized, and the medium is circulated from the bottom. It raises above, passes through the tube, and then it comes out. So when the medium passes through the tube, if it is going to be fluidized bed reactors. Now, coming to the advantage of the fluidized bed reactors, it generates a uniform mixing process. The uniform temperature maintenance is being maintained here, and the operation is also continuous. There is a minimum clogging process happening here. The next type of reactor that I want to discuss here is the photobioreactors. So in the photobioreactors, if you could see over here, mainly it is of two types are there. One is the outdoor or an open type photobioreactors. So as I've shown the pictures over here, so the open photobioreactors can be like artificial constructed lakes, or it can be performed like this in case of tubs, or these are the raceways in which the algae are constructed. So raceways are trenches that has been dug on the landforms in which the algae are being grown. So these kind of raceway system of algal growth, actually they grow the algae in this method to produce the biodiesel and also certain health supplements like astaxanthin and protein. And now coming on to the closed bioreactors. In closed bioreactors, as the name itself is suggesting that the entire process happens inside a closed system. So here we can see there are even biodegradable biopolymer plastic bags are also being used to grow the plant cells or the algal cells, which is actually going to produce the metabolites which we are needed of. Or it can be like these large tubes the algal also starts circulating, or it can be a static tubes like this. Long draft tubes can be constructed outside. The algal and the medium can be filled inside the draft tubes, and they can be completely exposed to the sunlight. See, in case of open door photobioreactors, there is always a chance of contamination. You cannot have a homogeneous organisms to produce the product. But where in case of closed photobioreactors, since the reactor is in a closed condition, we can always get the help of a homogeneous organisms in the product production. Now comes to the next type of reactors, that is reactor in series. So for to explain this, I've just taken a simple example of sewage treatment process. So in the sewage treatment plant, we have to degrade the waste into the simple forms or non hazardous forms. So in this process, so when we talk about the waste, so waste can be anything. There are different types of waste are there. So in this entire process, we need to degrade all these waste. So for which we need a specific bioreactor system in which a specific microorganism is grown, which is involved in the specific waste material degradation. So from the first bioreactor, the material is transferred to the second bioreactor where the parameters, the environmental conditions can be different. The type of organism sometimes can be different or it can be retained the same. But this, due to the alteration in the physical parameters, means it can be a change in the pH or in the temperature. What happens that is the microorganisms is able to degrade the second kind of a substrate in the second bioreactor. So, in when series of bioreactors are connected, in each and every part of that bioreactor, the microorganism can be degraded and then we'll move on to another type of bioreactor in series, that is the membrane bioreactors, which is most commonly used in nowadays in the wastewater treatment process. So when you talk about the membrane bioreactors, based upon the internal membrane bioreactor or the external membrane bioreactor. So when it comes to the external membrane bioreactor, the entire bioprocess, that is the waste degradation is done, and for the final waste degradation alone, the material, the medium is used biomembrane. So here the biomembrane is present outside this entire series of bioreactors. Whereas in the internal membrane bioreactors, the membrane bioreactor, it's an integral part of this entire series of bioreactors. So this is what is the main difference between the internal and the external membrane bioreactors. In the internal bioreactors, it can be an enzyme or an organism being immobilized, which is involved in the degradation of the waste into simple non-hazardous substances. So the advantage of this membrane bioreactors in sewage treatment processes, the sedimentation of sludge can be overcome. The biomembrane itself can act like a disinfection barrier because it has got a filtration process, and then the waste generation is greatly reduced. Use of membrane can be done very effectively. But the main disadvantage is that is membranes are very costly and it has to be replaced for every 5 to 10 years. And I've just shown you a picture of an industrial fermenters. They are huge fermenters and they are all present as an integrated systems, and they're all controlled by the external PLC control units. Now moving on to the bioreactors, which are helpful for the culture of the animal cells or animal tissue constructs. So before going into the fermentar, I would just give you a small outline about the animal cell line culture here. So the small animal cell line culture consists of hello. Okay, I'm sorry.
Ma'am, go ahead.
I'm just continuing with the bioreactors involved in the culture of the animal cell or for the animal tissue culture. So here, whatever cells we want to grow, animal cells we want to grow, that specific tissue is isolated, and then the cells are grown in a small volume in a tea flask. And after the specific cells are grown in the tea flask, then they are put into a scaffold or a carrier material, and then it is put into a fermentar bioreactor, like this bioreactor, which is around 250 ml capacity, so that the different type of cells, when it is linked through a scaffold, they get connected with each other, and then they get converted into a tissue. So these are small stirred bioreactors that are involved or used widely for the construction of the tissues. So this is a kind of artificial heart which is constructed from the rat cell, mice cells, and the person who has done this is her name is called as Doris. And here they have grown the inner cloaca of the ear also. And this is a hollow fiber bioreactor, which is mostly involved in the culture of the hybridoma cells, and it is involved in the production of the monoclonal antibodies. And whenever we want to go in for animal cell scaling up process, such small strip type of bioreactors are connected as a multi setup, and here the cells are cultured and the scaling up of animal cells are done. See, mostly for the cell line therapies or a stem cell therapy, the stem cells that we collect from the sample will be very, very minute concentration. So we need to perform these kind of bioreactions, or we need to scale up the cells, animal cells in the bioreactors. And now coming to the tissue construct, these are all different type of bioreactors that are involved in the tissue construct. So here they have just grown the rabbit tendons. So this is a first step for us to. It's a good thing for the all the sports injured people. So in a sports injury, the tendons are cut, and a particular part of the organ, like a hand or a finger or a leg or a toe, we cannot move them. So in that case, from the patient's body, the tendon cells can be collected, and then it can be grown in these specialized bioreactors. See, tendons are the tissues that are mainly having the contraction and relaxation property, so that the hand or the legs or the fingers we are able to move. So this is a function of the tendons that are being collected. And similarly, in this particular bioreactor, the cells are grown in the pressure condition. So when the cells are grown in the special condition, they will obtain their specific property as an in vivo in the in vitro also. So when such tendon cells are cultured in such a way, when they are transplanted into the patient, in the patient also, this tissue, since it is from their own origin, they will not experience any kind of immunological rejections. It will be accepted, and the tendon tissue will also be able to perform the contraction and relaxation inside the patient body also. And here I've shown you a picture of the heart pacemakers being constructed. So this is a chondrocyte cells. It's an individual chondrocyte cell, and these cells are being cultured within a scaffold. So the entire scaffold tissues, you can see it is contracting and relaxing, and this is how the heart patch tissues are being constructed nowadays. And such tissues, when they are replaced into the patient by the transplantation, no kind of immunological rejections can be experienced. And not alone that, we do have still type bioreactors which supports the growth of the meat tissues, and these meat tissues are lean tissues, that is less amount of cholesterol or fats, and they can be very easily consumed. So in this type, they have the specialized scaffolds on which the cells are being cultured, and when the cells in the scaffold, when they attach and they grow, they get converted into a 3D tissue structure. And this can be sold in the market and it can be consumed. And the next comes is the hollow fiber reactors that I was discussing earlier. So when you look at the cross section of the hollow fiber reactor, you can see there are hollow fibers, and on these hollow fibers, the cells are being placed. That is, the hybridoma cells are placed, and then when the medium is allowed to come in contact with the cells, that is the hybridoma cells, take up the medium, they produce the antibodies, and these antibodies can be collected from the spent medium from this reactor. So this is a small reactor, but it is able to produce so much of high value products. Now coming to the parts of the fermenter, before that I just want to give you a small introduction about the simple setup of a fermenter. So if you look at it, it need not to be a large fermenters, it can be even simple setups like this Erlenmeyer flask itself. So on the lid, whatever we need to add, all those ports can be attached, and not alone that, whatever liquid is taken out from the fermenter, those ports can also be attached over here. So a design of a fermenter is sensors. And now coming to the simple setup of a stirred type fermentar. So there is going to be a shell or the fermentar vessel, and the outermost of the vessel is going to be the cooling jacket where the cool water will be circulated so that the entire fermentation process temperature will be decreased whenever needed. And then it will also have the sampling port. It will also have the sterile port through which the medium or the specific microbial inoculum can be added inside the fermentar. And then, since it's a stepped type of fermentar, an agitator with the impeller blades is being connected. And not alone that, it also should have the various probes like the pH monitors and the temperature probes over here. And then at the bottom there is an orifice or a sparger through which the sterile air is pumped inside this fermenter. Now this is going to be a typical layout of the fermenter, what I discussed before, and this is an industrial setup of a 50 liter of stirred type of fermenter. Now coming to the components and the functions of each and every component of the fermentar is the fermentar. Let's start with the top plate of the fermentar. It is nothing but like a lid and it is made up of the stainless steel. Next comes the clamp. So the clamp main function is to have the top plate compressed into the vessel using the clamp. Then comes the seal. The main function of the seal is to prevent the leakage of the gas or the medium from the fermentar to the external environment. Next comes is a vessel. The vessel, as I discussed before, it should be made up of a glass or a stainless steel. It should be covered with a water jacket system, and then the vessel must also have various ports for the input and for the output analysis. Then there must be a dry port, which is mainly important for in case of the step type of fermentar, which supports the mixing up of the organisms and the medium and the gas. And next comes the impeller, which is associated with the agitator for the mixing purpose. And then we have the sparges. Through the sparges, the sterile air is pumped inside the fermentar. This is very important in case of the airlet fermentars and also the stirred type of fermentars. And next is the baffle. So baffle along with the impeller blades, it helps in the homogenization, mixing of the medium organisms and the gas. And the next comes is the exist gas coolers and the inoculum needles through which the microbial cells can be inoculated, or from the medium can be taken from the fermentar to analyze the concentration of the cells. And so this how the external ports are being set up for a fermentar. So these are all the external mediums that we have to, the liquids that we have to add inside the fermentar. It can be the medium. So medium means in a batch or in a fed batch fermentation process, the medium has to be added now and then. So for that we have to have the medium always ready in a sterile form. So whenever needed, the respective concentration of medium can be added into this fermentar vessel, or the inoculum must be kept ready as a working stock. So whenever needed, the cells can also be added into the fermentar, or it can be the acid or the base solutions which are added to the fermentar to maintain the pH during this fermentation process. So all these solutions can be either manually added or we can connect it with the computer software program. So whenever there is a need or a change in this pH, or when there is a need in the inoculum or in the medium addition, automatically from this respective sterile bottles, the respective solutions are added into the medium. And next comes is the feed pumps, which is a very important part of the fermentar. So the feed pump's main function is to transfer the medium or the nutrients or the microbial suspensions into the medium. And this can be done, the additions of the medium nutrients or the organisms can also be done with the peristaltic pumps. And then we do have syringe pumps, which helps us to manually add all the above said solutions to the fermentar. And next comes is the gas analysis. So in case of the gas analysis, either the carbon dioxide analyzer, oxygen analyzer, or it can also be connected with the mass spectroscopy. And then there is a sample pipe through which the sample can be drawn from the fermenter to check the concentration of the medium components, or to understand the the stage in which the microbial cell is present, either it is in a log phase or in a lag phase or in a stationary phase. And it should also be having the three-way inlet systems to insert the three different probes, where in case the temperature or the pH or the dissolved oxygen concentration can be monitored inside the fermentation, inside the fermenter. Now coming to the monitoring and the control parts of the fermentar. So the first thing is the temperature sensor, which is a platinum resistant electrode, or it can be a foam probe, which is kept above the medium level to sense the foam or to monitor the dissolved oxygen level. Heating pad directly helps us to heat the medium in order to maintain the temperature. Cold finger is also used to decrease the temperature to maintain the optimum temperature for the microorganisms growth, which is involved in the product production. And then we do have the rotameter, which helps in the controlling of the airflow inside the fermentar. The air pump actually supplies the air to the fermentar, which is nothing but our compressor or a simple small air pumps. And then the third thing is going to be, the last thing is going to be the peristatic pump, which actually helps us to pump in the medium, acid, base, or an antifoam agent directly into the fermentar whenever it is required. And now coming to the body, as I told you, it can be a simple glass vessel, as we all have it in our college, or it can be an industrial type of fermentar where the body of the vessel is made up of stainless steel. And these are the sealings or the gaskets that I was talking about. The sealings can be a gasket, like how we find the rubber tubing in our normal cooker, or lip seal can be present, or a O-ring can be present. But all these ceilings must be made up of a very sturdy fabric, which can withstand the pressure, which can withstand the heat. So they are normally made up of nitrile or butyl rubbers. And next comes is the aeration system. Coming to the sparger, which is very important in case of stir-type fermentar, and also for the bubble column airlit fermenters, and also the bubble column fermenters. So this is first type of aeration sparger is the porous sparger, which is an elongated tube, and around this tube we do have small holes. So when the air is pumped, the air is distributed into the medium in form of small uniform bubbles. So the bubbles comes in a huge force, and since they come in a huge force, they raise up completely with the length of the fermentar, and when the air rises up, it also helps in the mixing process. And next comes is the orifice sparges. So these are all the different pictures of the orifice sparges that I have shown over here. So it can be a spider sparger, or it can be a concentric ring sparges, or it can be a perforated disc shaped sparges. So in all these sparges, any one of the sparges can be very successfully used in the bubble column reactors. Next comes is a nozzle sparger. So nozzle sparger So it can be like this jet nozzle type of sparger, diffuser type of sparger, or perforated plate of sparger. Or it can also be a water jet sparger, which is mostly present in the fermenters, which are large in size, and the air has to rise to the complete length, and also maintains the mixing of homogeneous mixing of the medium, microorganism, and the gas. Now coming to the mixing process that happens in the fermentar, there are two type of mixing process must happen. It must be both axial mixing and also the radial mixing. That means it must be mixing the material content of the fermentar both in the vertical process and also in the horizontal process. For this, we require the baffles and the different type of impellers. So these are the pictures of the few impellers that are shown over here, and these are all the pictures of the impellers that I've shown here. The main function of the impeller blades or the stirring up process is to accomplish two of these things. That is, it has to mix the gas bubbles throughout the liquid culture medium, and it also mixes the microbial cells throughout the liquid medium. In this way, stirring ensures the uniform access of the microbial cells to the nutrient. And the impellers can be made up of stainless steel material, or it can be made up of this aluminium material also. And in most of the steady type of fermenters, the Ruston impellers are being used widely. And this is a pitch blade fermenters, pitch blade impellers over here. And for a very large stirred type fermenters, these kind of billet impeller blades can be used. And for the radial flow, these kind of bent impellers can be used. So whenever we are growing cells like fungal cells, agitators will be having impellers with the bent impellers, which helps in the radial mixing process. And next comes is the baffle. So in this picture, you can see baffle is nothing but stainless steel rods associated at the sides of the fermentar. So this allows the gas to and horizontal mixing, and how the baffle helps in a vertical mixing is being described in this picture. Now coming to the fermentar feed pumps. So the feed pump's main function is to supply the any one of the four materials that I'm suggesting now. It can be a medium, current, then the peristaltic pump gets switched off, and then it comes to a horizontal gauge structure, so that the flow of the fluid within the tubes are being stopped. And next comes is the syringe feeding system. In the syringe feeding system, the mechanical feeding of the medium or the buffer solutions or the antifoaming agents are done with the help of the syringe feeding systems. So in the syringe feeding systems, the whatever we want to supply to the fermentar is taken in syringe barrel, and then it is attached to the membrane disc like this, and the membrane disc with the silicon tubing is being connected to the fermentar. So when the medium is released through the syringe, it comes in contact with the membrane filter, where the sterilization process happens, and the biological medium are directly released into the fermentar. And these are all the syringe drivers which supports the filtration process of small volume of fluids to large volume of fluids. And now coming to the ports. Ports are very important in case of fermenters, because ports actually helps us to connect the external environment with the internal environment of the fermenter. So whenever we want to transfer any medium or a microorganism, suspensions, or any one of these ports. So port means temporarily it will be sealed. Whenever we needed, the opening can be created, and then we can release the material inside our fermenter. So these are all the needle inoculum ports to which the inoculum microbial cell suspension can be introduced into the fermenter. And this is a septum port, and this is a blind port. So septum port means it will be having a silicon cascade septum like this. So just with the help of a syringe, we can directly insert the needle of the syringe into the silicon port, and we can release the medium directly inside the fermenter. In case of blind port, whenever we don't want to use a kind of a sensor, then we can remove the sensor for the fermentar, and to fill the gap we'll be using the blind spotter. Whenever the probe has to be reintroduced into the fermentar, the blind spotter can be removed, and then the specific probe can be inserted. And this is a bypass sampler. Just like how we connect two water pipes together, we use the septum to connect the silicon tubings together. And this is also a connector coupling which connects different pipes of the fermentar. Like for example, we want the hot water to come and get itself circulated within the outside the fermentar jacket means, then the hot water pipe has to be connected to the fermentar with the help of the connecting coupling. Now comes is the valve. See, valve's main function of a valve is to allow the flow of any fluid fixed unidirectional motion. And there are different types of valves are there. So the different types of valves will be as open closing valve, drill spear valve, disc rotating valve, rubber diaphragm valves. So I'll just go one by one. So when we see the ball valve or the gate valve, it is mainly involved in the on-off control system. Butterfly valve is it is used for the large pipes, especially when we want to drain out the medium from the fermentar. At that point, you can see there will be a butterfly valve. So it will look like this. And then we have a check valve. The check valve's main function is to check the unidirectional flow of the fluids. And then comes the diaphragm valve. So the diaphragm valve is present for the main thing is for the stability purpose, as it is shown on this picture. The globe valve helps us again in the flow regulation process. The needle valve, slow pressure, it helps in the slow pressure release. And next comes is the plug valve. So the plug valve is a disc-shaped valve which is also connected with the on-off process of the valves. Then comes is the pinch valve, which comes as a diaphragm, rubber diaphragm valve. So the pinch valve's main function is to regulate and stop the fluid flow. It is otherwise called as a clamp valve. Then the check valves, as I told you, it helps us to check the flow of the fluid. So one example of the check valve is the airlock valve bulber. Next comes is the pressure control valve. The main function of the pressure control valve is to protect the entire fermentar system. When there is some mistake happens during the fermentation process, there is an excess of pressure being built up. Then in order to protect the entire fermentar systems, we do have the pressure valve. So the pressure valve will automatically collapse so that the extra pressure is accepted by the valve, and the valve undergoes a destruction, and the fermentar will be safe. And next comes is the safety valves. So safety valve can be the bar adjustable valve, which is to helps us to release the excess of gas from the fermentar to the external environment. In certain cases, carbon dioxide can be produced at a very fast rate. So we need to release the carbon dioxide. For that, the bar adjustable safety valve is necessary. And next comes is the stream traps. So stream traps are also one of the important component of a fermentar. See, when you want to heat up the medium, or in order to increase the medium of our fermentar, we supply the hot, hot water vapors are passed into the pipes. And then when the optimum temperature of the fermentar is being achieved, then the cold water will be circulated. In the pipes, some kind of unused water vapors will be trapped. So on passing the cool air, they might help in the condensation of these water vapors, unused water vapors, and the water droplets are getting collected in the pipes, and they might create contamination of the fermentation process. See, in case of pharmaceutical components, product productions, which has to be happening in a complete sterile environment, the stream taps are very, very important. The most important trap is the kinetic effect of fluid motion stream traps. It is a low density steam is flowing, it will be a high velocity. Likewise, high density steam flow means it will have a low velocity. The conversion of the pressure energy into the kinetic energy helps in the closing and the opening of the valve, as is shown in this picture. Now coming to the compressor. So for a very large fermenters, the air must be pumped inside the fermenter with a huge pressure, for which we'll be using the compressors. But for a small laboratory scale fermenters, air pumps are more than sufficient to pump in the air inside the fermenter unit. What if you are going to use the compressors must be oil free compressors, especially when the compressors are used to supply the air inside the fermenter, where the nutraceutical product production or pharmaceutical grade product productions are happening. Now coming to the detailed picture about the filters, we'll be using different type of filters. The first thing is about the membrane filters. So the membrane filters are cellulose acetate and nitrate with constant pore size of 0.2 millimeter to ensure the filtering of the particulate matter. And they are not cheap, but they are very costly. So whatever filters are shown over here, they are all the good examples of the membrane filters. Now come the packed bed filters. So in the packed bed filters, the column is internal of the column is stuffed with the cotton or the wool, and when the air is passed through the cotton or the wool, the filtration process happens. But the main disadvantage is that is, suppose the air comes with little amount of liquid in it, that is the hot water vapor passes. That means the cotton wool or the wool, it just gets compressed and it forms channels. So through the channels, when the channels are being formed, it is an indication for us to replace the internal part of the glass wool or the cotton wool. Otherwise, the air without filtration passes inside the fermenter and it can create the problem in during the fermentation process. Now comes the cartridge filter, as it is shown at the bottom one. So the cartridge filter, it is nothing but it can be a stainless steel or it can be a polycarbonate cartridge, and inside this the filters are present, and here this main function is to pump in the air and sterile the air, and they are very durable, but they are very expensive. Now coming to the most important part of the fermenter are the sensors. So sensors will help us to tell how what will be the exact pH during the product production or during the organism's growth. See, organism growth and product production, for which the optimum pH has to be maintained. So pH check must be done from the start day of the fermenter till the end day of the fermentation process. End day means the day when we are removing the liquid and starting our downstream process. So every moment we need to check our pH, the dissolved oxygen concentration present inside the fermenter with the help of these kind of dissolved oxygen sensors, or with the temperature probes can also be used to maintain the temperatures. Now coming to the gas analysis. So during the fermentation process, due to the metabolic reaction, greenhouse gases can also be generated from the fermenters, which has to be detected, and it has to be collected separately and made used for some other purpose. See, for example, during the fermentation process, we cannot it is going to contribute in the global warming. Detected by using the exist gas analysis, and then it can be stored inside the fermenter. And then it can be connected directly with the mass spectroscopy. The mass spectroscopy analysis can tell us during which period of fermenter what kind of gas is getting let out from the fermenter. And now coming, this is going to be the basic setup of a fermenter. So any person who's before you start a fermentation process, just make this kind of diagrammatic layout and make them ready. So this is going to be the basic setup of a fermenter. And now coming, this is going to be the other monitoring systems like gas analysis systems that is connected to the fermenter. So now coming, this is going to be the basic setup of a fermenter. So any person who's before you start a fermentation process, just make this kind of diagrammatic layout and make them ready. So then we, with the parts of the fermentation, what is going to be the other monitoring systems like gas analysis systems that is connected to the fermenter. And not alone that, this is called as a process program layout process. So in the process program layout, we can clearly have an understanding. Just looking out this diagram, you can understand what is going to be the process where is the solution? See, for example, it's going to be diagrammatic pictures. We have to first generate it before we start the fermentation process, because anything goes wrong, we will know in which pipe a problem has happened, or in which part of the fermenter a problem has happened, and where that part is connected to the fermenter. See, when we are discussing about fermenters theoretically, it is all clear, because here also you can see I told you blue color means alkali, or red color and orange color represents an acid. But when you look at a fermenter, it is just a stainless steel equipment connected with lots and lots of silicon tubings and steel pipes. You will not know whether through the steel pipe hot water is passed or cold water is passed. So in order to understand and identify parts of the fermenters perfectly before starting a fermentation process, these kind of layout pictures will be very, very helpful for us. Thank you all for your patience listening. Now we can go on for the question sessions. Hello? Hello?
Ma'am, your screen is not visible, ma'am.
No, now I've stopped sharing, sir.
You finished.
Yes, sir.
1 minute, 1 minute. Because there were some issues. At times it was like you went on mute. Okay, ma'am. Thank you very much,
Sir,
ma'am, for the. Yes, ma'am.
Sir, you have to read out the questions, sir, because I'm
Yes,
not
ma'am.
going live.
Yeah. Correct. No.
Yes.
There are some issues now. It's sorted out now, ma'am.
Yes, sir.
Okay. Thank you very much, Bharti ma'am, for the wonderful presentation. Now I would like to read out some of the questions raised by the participants. Ma'am, I find no questions, ma'am. Everyone has appreciated you and thanked you. Dear participants, if you have any questions pertaining to madam's presentation, you can ask so that ma'am will be able to answer your questions. No, ma'am, there are no questions, ma'am. It was too good, someone from Karnataka has told.
Thank you.
Ma'am, shall I tell the word of thanks or?
Okay, I think there is one question by Ganesh. Bioreactors used in agriculture feed.
Yeah,
Sir,
just now he's.
I'll just try to yes, sir. I'll just try to answer his question. Meantime, no, you can post the feedback links. Sir.
Sure, ma'am.
See, when you look into the fermenters that are commonly used in agriculture field, if you could see, there is one kind of fermenter that is now recently designed by Australia, and that is a actual in situ fermenter. That means on the land they do it for the degradation of the nitrogen waste that is coming out of the agriculture field. So in case of agriculture feed, lot of nitrogenous materials are being, chemicals are added to support or promote the plant growth. And these nitrate materials, when they get dissolved in the water, and they can contaminate the groundwater table. So in order to degrade this nitrogenous chemical fertilizers that they normally add it into the field, bioreactors are generated for them to degrade this nitrogen fertilizers.
And one more question
Ma'am,
from Surya.
how can we detect errors in the sensors? See, errors in the sensors, yes. See, if it is a fermenter or a bioreactor is just a new thing means, then we will not have any issues at all in the sensors. say, for example, we are working with a fermentar or a bioreactor continuously for a year or so. That time, sensors might have some problems. See, if it is going to be a pH sensor, means the pH sensor probe will be directly introduced into the fermentar medium. So there might be a deposition of salts over the sensors. So that time the sensors may not be able to detect properly the pH correctly. So what we have to do is now and then we have to remove the sensors and keep cleaning the sensors. And just by looking at the sensor itself, we will be able to see whether there is any deposition on the sensor or not. And if you are not sure during any process, say for example you are keep on using the sensor for more than 2 years of time period, means then you need to check the concentration of oxygen present inside the fermentar manually at least once. So if the sensor reading and what reading you have got manually is not correlating, then that means the sensor is erroneous and you need to replace it. Okay, I have a question by Ajay. If a parameter changes in the fermentar and we fail to notice it and notice it much later, there is no way to reserve it. See, if it is going to be a normal process like a degradation process, yes, we can reserve it. But just imagine you're going ahead with the pharmaceutical grade product production and you fail to check whether the air that is getting pumped into the fermentar is filtered or not. Then the entire process is going to become waste only. See, for that is what is the main role of a technician, is to check every step in a ferment during the fermentation process. Nowadays, in case of fermenters, it has been supported by bar 7 front of a fermenter and keep checking everything. By the software itself, the probes are will be activated. The probes will detect the concentration of the medium or concentration of the gas or what type of gas can be detected, and then the reports will be generated in your computer system. You just need to go access the database and get the needed data. See, for all these, the fermenter related programs, they are all codes are written based on the Python language. Okay, Mr. Amal Raj has asked a question like fridge. Is there any possibility of having fermenter in every house in your future? How can we make this happen? Amal sir, we already have fermenters in our house. That is the glass bottles in which pickling process will always happen. If you need for any kind of they do produce wine at home, they do produce beer at home. All these food productions need not to be supported with a sophisticated fermenters. And as you say, see, for example, we need to produce a pharmaceutical product in our house. Yes, we need a specific sterile environment, and we need to do lots and lots of innovations in this angle in order to give every household and correct literacy to person to handle a fermentar even at our house itself. There is a question by Madan Kulkarni. How can I get rid of zinc sulfate precipitation in LB broth or nutrient higher than 100 ppm? Sir, you need to do a titration process to detect the zinc sulfate concentration. And zinc sulfate, since it is a salt, you can add some kind of precipitation flocculation solutions in order to remove the excess of zinc sulfate. Or you can also go in for reverse osmosis, which is a ion mediated process to remove your zinc sulfate from your medium. Ajay has posted advantages of continuous culture over the fed batch culture, vice versa. See, when we talk about the types of mode of culturing of the microorganism, either it is to be adapted as a continuous process or a fed batch process or a batch process, purely depends upon and the type of products that we are interested in. See, there is no fixed system that for this particular product production we have to use only fed batch, or for this product production we have to use only this particular process. See, when you go for the wine production, we have to go on only alternative for the wine production. It has to go on only for a batch process. How can we prevent? There is another question: how to prevent the microbial contamination in plant tissue culture and in cell culture? Sir, a simple thing is when you want to grow a callus propagation in your medium, you always have a slant medium. On the top surface of the slant, you can add some little pinch of activated carbon to it, so that it will be removing the microbial contaminants from your medium. And sir, in case of plant and animal cell line culture, if cells are contaminated, it is better to change the entire batch, because we are going to culture the plant cells and the animal cells to get secondary metabolite products, which we are going to use it as a therapeutic or a diagnostic purpose, or like a nutraceutical components. So in that case, let's not take any chance of its contamination. Ma'am, is there any lifespan for an ordinary fermentar? See, a fermentar, as my knowledge is concerned, is just like a system. We can keep on changing the hardware and keep updating the computer system. Similarly, the main body of the fermentar can be maintained, but whereas all the probes, the peristaltic pumps, the dissolved oxygen ports, everything we can keep designing and changing according to our needs. So when you talk about the lifespan of the fermentar, as long as the inner surface of the fermentar is not having any kind of salt deposits, you can go very well with handling the fermentars. Whereas the probes, for every 6 months you change the probe. That is my advice to you.
And that's all, ma'am. I think all the questions you have answered. Yes. I will request our Bharti madam to convey the vote of thanks and conclude the session. Ma'am, please.
I take an heartfelt thanks to render my heartfelt thanks to the management of Guru Nanak College for giving all of us a platform as a department to organize this webinar series, and also giving me a chance to be a speaker in this technical session of fermentation technology. And I also like to thank all the participants who have participated in patient listening and asking clarifications during the session. I also thank my entire team of biotechnology faculty members in rendering their support, and also my dear students. My heartfelt thanks to all of you. Thank you.
Dear participants, the feedback for the session 4 have been posted in the chat box. Kindly submit your feedback. On request, we have posted the feedback link for the morning session, that is session 3 also. Kindly submit your feedback, and we shall meet tomorrow for the 5th session of the webinar series on fermentation technology. The resource person is Dr. Veena Gayatri, Assistant Professor at Department of Biotechnology, Stella Maris College. The link for the same will be sent to you by email and WhatsApp. Kindly join us and get benefited. Thank you very much for your wonderful participation.